Presently, we're at 74 species from 181 seed sources, but if somebody comes to us and they're like, can you grow that? The answer is yes, we will figure it out. We will grow it for you. So I am the only horticulturist here on the staff. Horticulture science is specifically the study of plants in an artificial environment, more or less. And whether that artificial environment is a greenhouse, tomatoes or trees, or your yard, which to a lesser extent is also artificial. My role here is to provide the aspect of horticulture science in a forestry industry. This process in the greenhouse is obviously an artificial environment, and I don't think anyone would argue that, between the absolutely artificial media and the artificial lighting, which we use in the springtime and the summer growing season to keep a steady 12-hour day length. Certain species are very day length dependent and will stop growing if that day length goes below a certain number of hours. So if you're growing that plant in the early spring when the day hasn't gotten long enough yet, you're going to get a very short crop. Even out in the field, I would consider that to be an artificial process, because even the sowing rate is calculated to manage the plant's growth. So how many trees you put down per linear foot is going to control how tall that plant is able to get, because it's competing with its neighbors for available resources, which we're also controlling. The mission's always been the same: to provide the highest quality, locally adapted plants for conservation and restoration purposes in Montana. And we're not so much concerned with the part of the plant that's above the ground. That's what all of our customers look at, because they don't know plants very well. What we're concerned with is root development. We want this gorgeous plug to hold together. We want all those roots. It doesn't matter if this top is this tall, which is about 7 or 8 inches, or 3 inches. As long as this is filled out and good, that is a good tree, and it's going to survive if you take care of it properly. But people always like this big top. So we kind of have two sets of parameters that we're looking at. We want that plug to fill out, and we want to get it to the customer's expectation of height. So who do we provide those for? They're private landowners, conservation districts, state and federal agencies, tribal agencies, conservation groups and organizations, the forestry industry, and any individual or group implementing conservation practices. So what that can mean is, if wildfire burns through your property, you want to replant, you come to us. If you built your house and you want to replant, you come to us. Stream bed restoration, all that kind of stuff, we provide those plants. The plant nursery used to be funded by the state because we are a state entity. We exist under the Forestry Bureau, Forestry Division, the Forestry Assistance Bureau. We function autonomously, but we still report to the state. So we used to get big time funding, $100,000 a year, I think, from the legislature. They discontinued the subsidy but kept us under their umbrella. So basically we have to follow all their rules, but we don't get any of the benefits. This seems very simple, but it's a lot of art. So I have to manage this crop so that it gets enough water, but not too much water, because if it gets too much water, the roots won't develop properly and they'll get root rot. But if I don't give it enough water, then I'll stunt the plants. So there's this rather fine balance. But there's a kinship. There's a kinship. The plants tell me what they need, and I just give it to them, and then the rest is useful neglect, and they just are happy. It's great. It's a good combination, me and them. I'm a simple person. I was raised simple. I'm never gonna build an Empire State Building. I'm never gonna have a monument built to me. But somewhere out there is a forest that I started in here, just like this little tiny forest. I've been here, it'll be 7 years in the spring, I think, and in that time, I think I roughly calculated I've put out about 5 million trees. So on some level, I've created something that's going to outlast me, which is cool, even though, you know, it's anonymous.
Everything looks really simple on paper in science, and then you have to realize that you're going down a path that no one's ever gone down before. And so you end up hitting a lot of dead ends, and you find out, you know, hindsight, oh man, that was a dead end, but you don't know until you go down it. So anyway, it's been a really fun project. Hopefully that paper will get out this year.
Borrelia burgdorferi is a bacteria, and it's in spirochete. And the cool thing I think about it, or you know, an unusual thing, is that it has a flat wave, almost corkscrew-looking shape, which is very distinctive of spirochetes. So the overall focus of the lab is to look at gene regulation in Borrelia. That is how Borrelia expresses its genes to cause disease and survive in a mammal or a tick. The way I would best describe that, or you know, relates to that, is that we grow the bacteria Borrelia burgdorferi in culture media in a liquid test tube, like I have right here. And you don't. So Borrelia doesn't live just freely in nature. It's always found either in a mammalian host, or actually in some lizards too. So in a vertebrate host or in a tick. You can't like pick up some pond water or any, you know, a piece of, you know, dirt and find Borrelia. Never been found there before. So it always is in a vertebrate host or tick, and here artificially we can grow it in liquid media, which really helps you study it.
So we're able to grow these things, and we can grow them if we need large amounts of it, you know, and we do sometimes, you know, that's the way to go, because you can't grow up that much in an animal. But if you really want to see how the environment is influencing the organism, then you need to do it in that environment. And I think it's kind of amazing that we can do those experiments here in Montana, where the ticks don't live. I mean, if those ticks were to escape, and they really can't, because we've got, like, you know, there, you know, multiple security around it, but they would just die pretty quickly out here.
Doing it in vitro, meaning in a container that's not in an organism. Definitely have to have the right incubator, the right CO2 level, the right temperature. The media was really key. The person who first isolated Borrelia burgdorferi, I think it was growing it in media, just trying all kinds of different things. And so what we grow it in is called BSK. It looks like cherry Kool-Aid, but it's like everything the cells need to grow and survive.
Alan Barber, who's now in Irvine, he worked really hard to grow them, and they're not easy to grow, and but it's an artificial thing, and it basically is like the kitchen sink. You know, they just kept throwing stuff into this until it grew, and they didn't just pick anything off the shelf. They thought about, you know, oh, this is you know, it's going to need this nutrient, and added this, and so we have this medium. It's called BSK for Alan Barber, Stoner, and Kelly, and basically it started with Kelly came up with this with a medium for growing it, and then it was modified by Stoner, and then that was modified by Barber.
You have to be creative here and kind of see the big picture, and a lot of troubleshooting, like solving problems. So every day you're confronted with something like, why does this look like this? This wasn't I expecting, and then to think about it. And I like the creativity of it. I like that every day is completely different. I'm pretty self-motivated, like my boss isn't breathing down my neck, so I can kind of write out what I'm going to do today, and I set my own goals and I do it. I really like that.
You know, we have a question, and then you get to determine how best to answer that question or ask that question and do the actual experiment. And you know, we don't do it by ourselves. It's a collaborative effort of everyone in the lab, you know, with Laura and Scott and myself and whoever else in the lab. So it's kind of fun and energizing to talk about it, and you know, you have a question, how are we going to answer it, come up with a way, get the data for it, and then see if that data answers the question.
It was only 12 below today when we got here. You know, You do occasionally have days where it's extremely cold and you're working outside, but you know they're going to come every now and again. Generally, it's pretty nice. I mean, and as long as you're prepared for it, it's not too bad. We have the Arley strain of rainbow trout here, and it's really important to our management programs throughout the state. We use it basically to provide angling in reservoirs and lakes. We don't plant any rainbow trout in streams or rivers. We just manage those for wild, self-sustaining populations. But we're able to provide angling with our annual fish plants into lakes and reservoirs with these fish. And they typically, what's nice about them is they typically don't reproduce in the wild. They grow really fast, so you can create a fishery very quickly with them. They're very easy to raise, so they're pretty friendly as far as having them around here. We have a lot of good success over the years with them. And they're aggressive feeders, so they're very catchable. So that's nice, anglers like that. This hatchery is designed to be a reliable egg source that our entire program can depend on from year to year, as opposed to going out in the wild and collecting eggs, and it's hit and miss every year. Well, here it's not. We have our Arley rainbow broodstock, captive broodstock, and we've had it since around '55. We provide those eggs. Well, we collect them, we fertilize them, we rear them to the eyed stage when they can be handled, and at that point, we assess the survival of each group of eggs. So we have an idea of how they're performing over time, and then they're shipped out to other production facilities that don't have a brood stock, and then they raise those fish to generally 4 inches, but sometimes bigger, and plant them around lakes and around their area. And then we keep about 200,000 to 300,000 of those eggs here that we do the same thing. You know, we plant lakes around this area.
You know, these fish start becoming ripe in the first week in October, and it goes in a bell curve. We start spawning, we start checking fish roughly the first or second week in October. First full week in October, we'll go down and run all the 3-year-old females and all the 4-year-old females through the spawn house, check them, see which ones are ripe, and it just goes in a bell curve. And you know, at the beginning of the season in October, you're not going to get a lot of fish, but you're getting a few. And towards the middle of the season, like you know, towards the middle of November, you're getting your most fish. And towards the end towards December, you're tapering off and not getting very many fish. So every day or every spawn week, on a Monday and Tuesday, we'll go down, we'll go through all the 3-year-old and 4-year-old females, check them, see if they're ripe. The ones that aren't ripe, we call them green, and they go back into the raceway that they were in before. If they're ripe, they go into a separate raceway, keeps them separate. And then the following Wednesday, the day after we've sorted all the fish, the ripes from the greens, we start spawning. The first thing in the morning, we'll take milk from the males, take individuals, and all the milk will go down to the spawn house, and then we start crowding in the females. We'll collect the eggs from 3 4-year-old females into one bucket, and we'll have the milk that we would have collected that morning, and we'll have 3 vials of milk, and we'll pool the milk, drop it on, stir it in with the eggs right there, add some water to help with fertilization, and mix them up just a little bit, and then that bucket of eggs will get set aside so that we can do the next group of fish. And we'll do them in 4 groups of 3. And then each time we do 3 fish in one bucket, they'll get set over on the counter. And once we get 12 done, we'll get fresh water for for the MS, and then each one of those buckets, all 4 of those buckets of eggs, will get rinsed out. You want to get rid of all the excess ovarian and excess milk, and just clean them off. Once they've been rinsed, then they go into a cooler to be brought up here. Air spawning is nice. I can't imagine how tough it was before air spawning when you had to hand spawn all these fish. But air spawning's nice because you got to anesthetize the fish, make sure they're out. It makes it very easy because it's very quick and physically easy on you, and it's also on the hatchery personnel, but it's also very easy on the fish. Instead of pressing against their body cavity, pressing against their internal organs, to get those eggs expressed out, you're just slipping a hypodermic needle inside the body cavity underneath the pelvic fin, and fills up the body cavity with oxygen and forces those eggs out through the vent. Really, the most important thing you got to do is make sure that you burp them. Make sure you get the majority of that air out. That way they don't float upside down when they do come out of their anesthesia. It just makes it a lot easier on us because you're not physically pushing on those fish and handling those fish. They're already big enough as it is. And then it's easier on the fish because you can express all those eggs out very quickly, and you're not pressing on their organs. Instead of pressing on them 20 times to get all the eggs out, you're just pressing on them 1 time to get the air out. So it's just a lot quicker too.
One of the things I find rewarding is when we have school groups here. So we have a lot of educational interaction with kids of various ages. So you get to at least pass on that message of aquatic conservation, environmental awareness, things like that are important, So that's one of the things. Certainly enjoy being able to provide a product that really provides a lot of entertainment and recreation throughout the state. There's a lot of happy fishermen out there just taking their spare time and going fishing and having a good time. So just knowing that is pretty nice too. Nice job, Stephanie.